Hi, this is Ritik, your biology subject matter expert from MEMONEET. So today in this video, we will be concentrating on the very important chapter that is the cell cycle and cell division. So what are the aspects that we are going to cover in this video is what do you mean by cell, then the cell cycle, the M phase, and the significance of the M phase, which is nothing but the mitosis. Okay. So we will be concentrating only with regards to mitosis in this video lecture. So first thing, very first. So what do you mean by cell and the very blooming? Okay. Kind of what do you say that? Uh, so excitement in the way like you will always find it question like how a single cell they join together, then how we all got emerged. So how exactly the cell division occur and how this all mechanism happens, how this cell they get converted to the tissues, then from a tissue you will actually absorb in what way the growth is and all, right? So from tissues to the organ to the organ system and the entire organisms. It could be any organisms. So how this organization is happening. So growth and reproduction are actually the characteristics of the cell. Indeed, all the living organisms, you will see this kind of thing in all the living organisms. So all cells, they reproduce by dividing into two. Like we have one single cell, this cell will be divided into two. Okay. With each parental cell giving rise to two daughter cell each time they divide. See, if this is a parental cell, it will give rise to two daughter cell. Again, these two daughter cell at certain peak, they become a parental cell. Again, if they divide, they'll give rise to the two daughter cell. So in this way, what happens? Cell division occurs. Okay. So, therefore, these newly formed daughter cells can themselves, that's what I was saying you, they can themselves grow and again divide, giving rise to a new cell population that is formed by the growth and division of single parental cell and its progeny. In other words, such cycle of growth and division allow a single cell to form a structural constituting of million cells as actually can be called as the cell division, cell cycle. So let us know it in a clear way what exactly the cell cycle is, how the division occurs. Division means if I consider this as a single cell, automatically it won't divide. There has to be certain mechanism, right, that goes. So what is it? Right. So here the cell division is a very important process in all the living organism. During the division of the cell, DNA it replicates and cell growth also takes place. That is, the cell growth takes place. Say like inside the cell, you will find the DNA and you will also see the DNA gets replicated. Equivalently, the cell growth also takes place. So all this process, that is the cell division that I'm talking about, DNA replication and the cell growth, right? Hence, have to take place in what way? In a very controlled and coordinate way. Because, see, cell division, DNA replication, cell growth, it has to happen in a very specific coordination way. So if this doesn't happen, it is going to affect in the cell division. So therefore, they have to take place in a coordinate way to ensure the correct division of the cell and formation of what? A progeny cell containing intact genome, that is specific correct genome. If not, it will lead to mutation, cancer, many biological, physiological disorders. The sequence of events by which a cell duplicates its genome, synthesizing the other constituents of the cell, and eventually dividing it into two daughter cells is termed as what? Cell cycle. Clear with what exactly the cell cycle is? Although the cell grows, okay, is a continuous process. That means the cell division, cell growth is a continuous process. DNA synthesis occurs only during one specific stage in the cell cycle. Cell division occurs, cells growth will take place, and it's a continuous process. But the DNA which is present in the cell, that it has to what? The synthesizing of the new DNA, replication and all, right? So that happens in one specific stage of the during the cell cycle. So what is the stage? Where exactly it happens? That comes a question in our mind. So keep that question because I'll be explaining you in the coming slide. So what happens here is the replicated chromosomes, that is now the DNA is replicated, are then distributed, okay, to the daughter nuclei by a complex series of events during a cell cycle. So first question that comes in my mind, your mind, everybody's mind is, in which stage does the DNA synthesize occur? Okay, now in any specific stage, I'll be discussing with stage. So now the DNA synthesis has been happened. So how does DNA synthesize? Like it will be, monitored to, divide them in an equal ratio to both the daughter cells. How does this happen? Is again a question. So there we are going to concentrate on phases of cell cycle. So a typical eukaryotic cell cycle is illustrated. Eukaryotic cell cycle is means human cell cycle. So if we culture it, the cell divides one and appropriately and within 24 hours. Every 24 hours, cell division can be observed. Okay. However, this duration of the cell cycle can vary from organisms to organisms. You need to note down this point. So in human being, every 24 hours, the cell division may be observed. But in other organism, it differs. Okay, and also from cell type to cell type also it may differ. Say for example, yeast can progress through the cell cycle in only about 20 minutes. Within 20 minutes, like it can progress. Then the cell cycle is divided into two basic phases. Can you name them? One is obviously the interphase, will be the first phase. Then we have M phase. Okay. So, which is M phase is nothing but the mitosis phase. So, we'll see this. So, the M phase or the mitosis phase, which represents the phase when the actual cell division or mitosis occurs, and the interface, right, represents the phase between two successive M phase. That means if the cell has completed the M phase, again it should undergo the M phase. So between two successive M phase, the middle phase is actually called as interphase. Interphase itself says it's a middle phase. Okay. So the it is significant to note that in the 24 hours average duration of the cell cycle of human cell division proper lasts for only about an hour. So that is the cell cycle completion of one cell cycle, it takes average 24 hours. But cell division, it occurs lasts just for 1 hour within this 24-hour cycle. So now the interface lasts more than 95% of the duration of the cell cycle. So out of cell cycle, 95% actually happens, interface takes place. So what about the other remaining 5%? The M phase starts with the nuclear division, then corresponding to the separation of the daughter chromosomes, which is actually called as separation of the daughter chromosomes is actually called as what? So again, an important question. It might feel very silly, but this is where we actually make lot of mistakes. Okay. So we need to know that is called as kinesis, and usually ends with the division of cytoplasm, which is called as cytokinesis. You should know what is karyokinesis and cytokinesis. Clear? So karyokinesis is something where the daughter chromosomes they divide, they get separated from each other, and usually if the cell cytoplasm gets separated, divides, right? We normally call it as the cytokinesis. Cool. So for next, the interphase through called the rising phase is the time during which the cell is prepared for the division by undergoing both cell growth and DNA replication in an orderly manner. That means now, after all the process, now again it is ready to undergo the division. Then it is actually called as interphase. So the interphase is divided into three future phases. Say, like, can you see this? This is the interphase, G1 phase. Then you will see the yes phase, and you're going to see the yes 2 phase. So where yes phase, right, you will be calling this yes phase as synthesis phase. Okay. Synthesis phase where the DNA synthesis takes place. Right. So once the synthesis and happens and all, now this is the M phase. So can you see out of the circle, most of this has been taken by what phase? Interface. So from here to here, right? So it is the interface where this portion is M phase, that is mitosis phase. So therefore, 95% has been taken by interface itself. Okay. Coming to the very first phase of the interface, that is the G1 phase. The G1 phase corresponds to the interval between the M phase and initiation of the DNA replication. Very important. So during this G1 phase, what happens is the cell is metabolically active and continuously they keep on growing. But in this phase, you won't see any replication of the DNA. Okay. So next is yes phase or synthesis phase is where it is actually the marked as the period during which the DNA replication takes place. Okay. So in G1 phase, you will observe only cell growth. You won't see the DNA replication. Then where exactly we are going to observe the DNA replication is in the synthesis phase. So during this time, the amount of DNA, okay, per cell, what happens, gets doubled. So if the initial amount of DNA is denoted as 2C, then what happens? It will get doubled. Then it is going to increase as 4C, right? Right. However, there is no increase in the chromosome number. So chromosome will be exactly the same. Okay. So we have 46 number of chromosomes. You'll see that itself. There will be no decrease or increase of the chromosome. Only thing that you're going to see is the DNA will be increased. Okay. So the cell had haploid or 2n number of chromosomes at G1. Even after S phase also, what happens? The number of chromosomes remains the same, that is 2n, deployed in nature itself. In animal cells, during the S phase, what happens? DNA replication begins in the nucleus and the centrioid duplicates in the cytoplasm. So then after S phase, we will move to the cell cycle will move to the G2 phase where the proteins are synthesized and preparation of the mitosis while cell growth continuity. Some cells in the adult animals do not appear to exhibit division. That is, say, like heart cells and all. Once they reach adult, right, they do not divide. There are certain cells which actually are restricted. So once it is done, that's fine. It again under it doesn't goes undergoes cell division, and many other cells divide only occasionally as needed to replace cells that have been lost due to some injury or cell death. Okay. So these cells which do not divide further exist in the G1 phase itself, and instead of entering into the yes phase, what they do is it is going to enter into the G naught stage, that is quiescent stage of the cell cycle, arrest stage, that is what we normally say. Cells in the stage remains metabolically active, but they will lose their replication cell growth, that is no longer proliferate unless cells on to do so depending on the requirements of the organisms. So in animals, the mitotic cell division is only seen in diploid somatic cells. However, there are few exceptions, okay, to this. When haploid cells divided by the mitosis, for example, we have seen haploid cells in male honeybee against this, and the plant can also show the mitosis division in both the haploid and diploid cells, N and 2N. Okay, for some examples are alternation of generation in plants, which you see, so they will also show this kind of cell division. Right. So that is a plant species and a stage at which the mitosis is seen as haploid cell. So I'll concentrate on what is this M phase, mitosis phase. So this is the most dramatic period of the cell cycle, involved a majority of recognition of the virtually all components of the cell. Since the number of the chromosomes in the parent and the progeny cell is the same, it is also called as equational division. So equally division. True for convenience, mitosis have been divided into four stages. One is stages of the nuclear division, that is the karyokinesis, yeah, the nuclear division or the DNA replica divisions, right? So it is very essential to understand that cell division is a progressive process and very clear cut line cannot be drawn between various stages. That is what we need to understand here. So this karyokinesis involves four stages. One is the prophase. Okay. Then we have the metaphase, then anaphase, and the telophase. At the end, you are going to see the cell division, daughter cells. If you say this as a parent cell. So first, we will concentrate. What do you mean by prophase? So totally prophase, metaphase, anaphase, and telophase. So prophase is which the first stage of the karyokinesis of the mitosis, followed by after S stage and G2 phase. What happens? You will first encounter what? The prophase. Okay. In the S and G2 phase, the new DNA molecules which have been formed, they are not distinct but intertwined. So like this. So that thread-like structure you will be observing. Prophase is marked by the initiation of the condensation of the chromosomal material. The chromosomal material, what happens? It becomes untangled during the process of chromatin condensation. Then we have the centrosome, which had undergone duplication during S phase of interphase. Now begins to move towards opposite poles of the cell. Can you observe here? They get condensed and now they start moving to the, so it is thread like. So now they are getting what? Condensed here and they started moving to the opposite pole. So the completion of this prophase can thus be marked by the what all the characteristics. One is the chromosomal material condensation to form a compact mitotic chromosome is one thing which you are going to mark. Then the chromosomes are seen to be composed of two chromatides attached together. So this is one chromatide and this is one chromatide attached together at a centromere. Then we have the centrosomes which have undergone duplication during the interphase begins to move towards the opposite poles of the cell. Each centrosome radiates out the microtubules called aster. So if they are the centrosoles, okay, so it is going to radiate the microtubules here. They have moved opposite poles now. This is what you are observing. So then they're called as asters, and these two asters together, they are going to form a spindle fibers here. Okay. Then the cells at the end of the prophase, were viewed under the microscope, do not show this Golgi complex, endoplasmic reticulum, nucleus, and nuclear envelope. Right. Then after the prophase, we will be seeing the metaphase. So after the prophase, what happens? The complete distinguishing of the nuclear envelope mark at the stage of the second phase of the mitosis. Hence, the chromosomes are spread throughout the cytoplasm of the cells. So now you can see this. After this, all spindle fibers are appeared. So the spindle fibers, the centrosomes that have been attached to it, right? So by the stage, what happened? The condensation of the chromosome is complete and they can be observed very clearly under the microscope. Thus, then, in the stage at which the morphology of the chromosomes is more easy to study, as you can see this, you can easily study them. Then at this stage, what happens? The metaphase chromosome is made up of two sister chromatides, which are held together by the centromer. So this white color that you are seeing are actually the centromer, which the sister chromatides that are held together by the centromer. Then you will see a small structure at the surface of the centromer. They are called as kinetochore. Okay. So these structures serves as a site of attachment of the spindle fiber. So here in the what do you say that chitinochore, the spindle fibers are attached. Okay, to the chromosomes that help in the momentum and its position. They'll keep in the proper position, then move its position at the center of the cell. So metaphase is characterized by the chromosomes coming to lie in an equatorial plate. So all the chromosomes will come and you will be seeing in an equatorial plate. So that is called as metaplate. Okay, metaphase chromosome. Then the plane of alignment of the chromosomes at metaphase is referred to as the metaphase plate. The key features of the metaphase are spindle fibers that attach to the kinetochore of the chromosome is one point. Then the chromosomes are moved to the spindle, equally, then get aligned along the metaphase plate through the spindle fibers to both the pole. After metaphase, you will see the anaphase. At this anaphase, what happens? Each chromosomes arranged at the metaphase, they will get split. So, like here, you will be seeing the arrangement. They'll get splitted as you are observing here. So then what happens simultaneously? The two daughter chromatides, now referred to as the daughter chromosomes of the future daughter nuclei, begin to migrate towards the so they homologous chromosomes they get divided to form daughter chromosomes and they'll move towards the opposite poles. So here these are moving here and this is moving here. So opposite poles. As each chromosomes move away from the this is an equatorial plate, right? So move away from the equatorial plate. Hence at the leading edge, how they are moving, this is the leading edge and whatever you are seeing, there are arms here. Okay. So thus the anaphase stage is characterized by the following key events. Say like the centromer splits and the chromatide separates one point. Then the chromatides move to the opposite pole as your second point. Right. So what you're going to see another very important thing is during the momentum itself, you're going to observe certain shapes say like I shape, L shape, J shape, and V shape of this chromosomes. Anyhow, then we will be seeing the last phase of the M phases, the telophase. At the beginning of the final stage of the karyokinesis, the telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosomes can no longer be seen. That means the way you have been seeing in the previous slide, you will not be able to see those chromosomes in a very clean way. Each set of the chromatin materials tends to collect at each of the two pole. So this is the stage which shows the what are the key events that you are going to observe in the telophase now. Chromosomes cluster at the opposite spindle poles and their identity is lost as the discrete elements. Then the nuclear envelope develops around the chromosomes cluster at each pole, forming two daughter nuclei. Can you see here? Then the nucleus, Golgi complex, and endoplasmic reticulum and all. So you can absorb them. So at the last, what happened? The cytokinesis will what you are going to see. So after the completion of all those four phases, now what happens? You will see that the mitosis accomplished not only the segregation of the duplicated chromosomes into daughter nuclei, but the cell itself is ready to divide into two daughter cell by the separation of the cytoplasm, which is actually called as cytokinesis at the end of the cell division gets completed. So once the cytokinesis will happen, then that is the end of the cell division. So in an animal cell, this is achieved by the appearance of furrow in the plasma membrane. Furrow is nothing but this thing, what you are seeing, right? So there actually the furrow gradually gets deepens and ultimately joins in the center, divide the cell cytoplasm into two. The plant cell, however, they're enclosed by the relatively intransible cell wall. Therefore they undergo the cytokinesis by different mechanism. Like in plant cell, what happens? The wall formation starts in the center of the cell, and later on division will be seen. Okay, then the formation of a new cell wall begins within the formation of the simple precursors. Then they're called as cell plate that represents the middle lamella between the walls of the two adjacent cells. So if it is a cell, what happens? So a middle wall will form. But here what happens? Instead, a furrow has been formed in animal cells. So in plant cells, you will be observing a middle wall, then the wall of two adjacent cells. At the side, at the time of the cytokinesis, what happened? The cytoplasm plasmic division, the organelles like mitochondria, plastids gets distributed between the two daughter cells. In some organisms, the karyokinesis is not followed by the cytokinase, as a result of which multinucleate conditions arise, leading to the formation of uh, cytosm, that is the liquid endosperm in coconut. That is, if you observe the coconut, okay, so that is the liquid endosperm, the liquid water, so that is what you're going to see. That means immediately after the karyokinesis, cytokinesis will not happen. So then all the nucleus, they won't have those cell wall and all. So you will see that in a liquid form. So, what are the major significance of the mitosis then? Why mitosis is very important? So, mitosis, or it is also called as equational cell division, is usually restricted to the diploid cell only. However, in some lower plants and in some social insects, haploid cells like honeybee also divide by the mitosis. So, it is very essential to understand the significance of the division of the life of an organism. Then the mitosis usually results in the production of the diploid daughter cell with identical genetic complement. The growth of multicellular organisms is due to mitosis. Cell growth result in like the cell growth results in the distributing the ratio between the nucleus and the cytoplasm, and therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio, which is a very significant contribution of the mitosis in cell repair. The cell of the upper layer of the epidermis, the cell of the lining of the gut region, blood cells are being constantly replaced. Okay, so in blood cells and all, you will be seeing the cell division. Mitosis cell division. So mitosis division in the meristematic tissues, the apical and the lateral cambrium result in the continuous growth of the plants throughout their life. Because of this mitotic division itself, you will be seeing the continuous growth of the plant throughout their life. So this is all about the significance of the mitosis. So in the coming video, we'll be concentrating more about what are meiosis and what are the types that comes under the meiosis and what is the significance of the meiosis. Till that, if you feel any doubt, you can reach me out to the comment section. And you can also, message me through the Telegram channel. Don't forget to subscribe, like, and also share with your friends. Thank you.